So hi friends, how are you doing? In this video, we'll be discussing on the central dogma of information pathway and the genetic expression. We'll be covering the objectives, the concept of central genetic information pathway, the classical versus modern central dogma, and the gene expression. Hope you will enjoy this content. So what is central dogma of genetic information? It was given by Francis Crick in 1950s, and it suggested that the genetic information is usually unidirectional. What does it mean? It means that it starts at a particular point and stops at a particular point. It starts with the DNA and it flows to ultimately to proteins where they are synthesized for their function and structure. And the middle molecule through which this genetic information flows is the RNA. As you can see in the diagram, the DNA synthesizes RNA, gives its information to RNA, and RNA in turn gives the information to proteins. So proteins are ultimately synthesized by the information given to them by RNA, and this information is actually stored in the DNA molecule. This is the unidirectional flow of genetic information. So what is central dogma of unidirectional flow of information? It includes usually three processes as envisaged by Francis Crick. First is the replication. This involves the synthesis of a new DNA molecule on a template DNA, on a parental DNA molecule. So the information is given by DNA to a new synthesizing DNA. Then there is a transcription, which means the information from the DNA is coded into the synthesis of an RNA molecule. So when DNA performs and makes RNA, it is called as transcription. And finally, translation, when proteins are synthesized on the information given to them by RNA molecule. These three processes are classically given by Francis in the classical model of the central dogma. That is, the information of the DNA is flown to RNA, then RNA gives the information to proteins. So there is a unidirectional flow from the DNA to the proteins. However, later on what was found is that this central and unidirectional flow of genetic information does not hold true universally, especially for certain viruses and certain modifications of certain genes where some other processes were also involved in addition to the three centralized processes as given by the Francis Crick. Hence, this central dogma was later modified, and what we now refer to as the central dogma is actually the modified version of the central dogma. And it includes, in addition to the three processes which I just told you, it also includes certain other major processes as well, which are particularly here as listed. First of all is the reverse transcription. Reverse transcription is a process where an RNA is utilized to synthesize DNA. That is, it means that RNA can go back and synthesize DNA on its own template. This is usually seen in RNA reverse transcription viruses, where they are able to synthesize the DNA segment from the RNA which they contain as a structural unit. A second process is the RNA replication. RNA replication is similar to the DNA replication. However, here RNA acts as a template to synthesize the new RNA as well. So RNA can synthesize RNA as well. Then there is another process of RNA editing as well. RNA editing is utilized in certain specialized cells where the RNA after synthesis is modified, edited into a mature RNA. So this process is also added to this central dogma. And finally, the concept of prions. Prions are the specialized proteins which are mutated as found in certain scrapie disease or what is called as Kuru disease, where the proteins act as a scaffold to direct their synthesis and or the folding of a protein into a variant form of a protein which is having a function less or no function at all. So all these processes were added to the central dogma so as to modify it in a current version which is not unidirectional, which can move in both directions, whether the DNA makes an RNA or RNA can make a DNA. Similarly, the RNA can make its own copies of RNA as well. and it can go editing as well, and some specialized conditions where proteins can direct the folding of the proteins as well through prions. So this is the version which I already told you in a different graphical manner. Last but not the least, talking about the concept of gene expression in a viable cell. How does a cell make itself viable? It does so by following the central dogma, the three main processes of the central dogma. That is that its own DNA synthesizes its DNA copies furthermore through replication, or it undergoes a transcription to make RNA, and RNA in turn translates proteins. And proteins are the dynamic and structural units which make cell viable, not only structurally but metabolically as well. But in eukaryotes, this is not pretty much it. This is not as simple as seen in this slide. Why? Because each process of the central dogma is either followed by pre-modifications or post-modifications, and sometimes also co-modifications. So each process has actually three sub-phases. That is the pre-process, co-process, and post process. For example, in case of an RNA, it goes for pre-transcription, co-transcription, and post-transcriptional modifications to make RNA viable structurally as well as functionally. Similarly for the proteins, and this is most importantly for RNA viability and function, as well as for protein function and structure and folding. So each process in eukaryotes is usually followed by post-synthetic or co-synthetic or pre-synthetic modifications, particularly in RNA and proteins. So well, this is pretty much it about this topic. I hope you enjoyed the content and it was beneficial to you. If you like this videos of mine, kindly subscribe to the channel. I will be uploading more of the videos for molecular biology and rest of the biochemistry soon. Till then, see you. Take care of yourself.